So Dr. John Thornback will be giving us the presentation on the overview of SS656. So Dr. John Thornback is the Chief Operating Officer of the Diagnostic Development Hub Singapore, and he has over 30 years of experience in diagnostic industry. So he was initially the head of R&D and then sales and marketing, and he's the CEO of various startups and spin-offs in Canada, UK, and Singapore. So Dr. John Thornback, please.
Thank you, Young Hai. Okay, can I have the first slide, please? Okay, well, microRNA-based diagnostic development has been a major focus for the Diagnostic Development Hub since we started. But Patrick and I, who were on the committee to help develop this standard, are only the front of a team of people within Diagnostics Development Hub who have been involved in the development of this standard. In particular, Dominic, Anisha and Janice, who were involved in the biomarker discovery, and Felicia and Xiaoyun in the verification and validation steps as well. And Jessica from our regulatory group also helped with the administration of the committee putting forward for developing this standard. So they're the people who actually did the work. I'm just the front person. Okay, so why do we need a standard now, and what is actually in the standard? That's the message. So why RNA? Well, microRNA. Well, it's a master regulator. It's been found over the last, 20 years almost now since it was discovered in humans. We're getting a growing knowledge of what it's implicated in. There is, it's been shown to be a master regulator of endogenous gene expression. There's growing clinical evidence that it can be used in, that it's implicated in lots of different disease states. And importantly, it has exceptional stability because it's incorporated generally inside vesicles. So it has all the very good characteristics of a good biomarker. And we're also seeing now that industry is beginning to take up microRNA. Rexus, who you'll hear from soon, Lehan is giving the next talk, I believe. We'll tell you a lot more about what Rexus has done. But there are a number of companies now who are involved in, who've taken the concept of microRNA and developed diagnostics-related products. So basically, this is, I guess, gene expression 101 for those of you who've forgotten your basic biology. You have DNA, then basically you get a single strand of that DNA is transcribed into messenger RNA. That messenger RNA then moves from the nucleus into the cytoplasm and is then taken up by ribosomes who look at the codons and then form amino acid chains and then form the proteins. That's very simply what happens. So where does microRNA come into this? Well, microRNA comes into this because basically it regulates it. There is a microRNA gene that sits in the DNA, and what it does is that it interacts, the microRNAs react with the messenger RNA to block either the translation of the message or it degrades the messenger. So it has two main functions. What's interesting is that in some cases, the excretion that the microRNA can be excreted into biofluids. For instance, in tumor cells naturally excrete vesicles which include microRNA, and then they are transported to other sites where they block other messenger RNA. So there's a lot of potential diagnostics involved in that. But it can happen that if there is damage to tissue, such as in cardiac situations, then the microRNA is released in damaged tissue. So there are multiple possible applications. As I said, therefore, microRNA regulates a lot of different physiological processes. Cancer is the one that we'll hear a lot more about, but it's also involved in cell proliferation, cell differentiation, where it degrades the target messenger RNA. and also in inhibition of translation, things like apoptosis and things like that. So because of that, microRNA dysregulation is associated with numerous different pathological states, which means that actually there is a possibility of developing microRNA-based diagnostics for many different potential indications. In the non-cancerous area, it's been implicated in everything from acute myocardial infarction to rheumatoid arthritis. In cancers, it's been shown that it's implicated in lots of different cancers. The first one you'll hear about today, I think is going to be in gastric cancer. And so, what we're looking at is the circulating microRNA profiles. Okay, so to summarize, it's an active regulator and reflects numerous processes. It's a minimally invasive test because you're taking it from a biofluid, typically serum or plasma. And it's also also measurable using existing platforms, although we have had new technologies developed. So it can be used, microRNA-based diagnostics potentially could be used as diagnostic, prognostic, or predictive biomarkers. So there's a potentially wide application for these products. So what are the challenges then? MicroRNA are very small pieces of RNA, 18 to 22 nucleotides. And the RNA, the microRNAs differentiate sometimes by only one nucleotide. So, and also because, as I said, they're buried inside biomolecules. So that creates a lot of problems. And it's really been the development of new technologies, either the stem loop technology, which is illustrated here, or things like LNA, which have allowed us to be able to amplify the initial strand the initial strand RNA to give amplification to then develop the diagnostics. As it says there, finding a needle in a haystack. So what is included in the standard? Basically, it's a roadmap. And it takes you through the different stages of the process. The first stage is, of course, biomarker discovery. And in this, what you're looking at is trying to develop, to compare the expression of the different microRNA, of which there are about 600, and they're all in a database, between normal and patient populations, a patient population. So having done that, we screen the 600. You can do that by a number of different processes. You can do it by microarrays, or you can do it individually. You can screen each microarray individually. And you bring that down to a much smaller number. And then when you're doing the validation, you will then take different patient populations, different patient samples, depending on what population you want, whether that's a global population or it may be a population within a certain, say, Southeast Asian population, or it may be for a different, just a very small group of patients in certain diseases, and bring that down to between 12, 24, or 32. And if you take forward, say, 12 microRNAs, which are the ones that you find to have the biggest differentiation, then you go into the standard validation verification process that is carried out with normal PCR tests. And then final stage, you do a clinical validation, and then that will be a completely new set of samples. And that creates the documentation that will give you the clinical utility, which will give you the final product. So what does the scope include? It consists of design development, performance evaluation of the molecular diagnostic assays. It gives you guidance for the development process, and it's applicable to all microRNA-based diagnostic assays. So it's either a commercial IVD or assays that have been developed and used in clinical laboratories, i.e. LDTs, by another word. So thank you very much. I hope that's given you a brief introduction to what microRNA is and what's in the scope of this standard. And we hope that you will find it very useful.
Thank you, Dr. John Thornback.